DUX4 protein, as you've heard, is the protein that causes the pathology in FSHD, and its inappropriate expression in muscle is driving the disease. So as we formed Fulcrum Therapeutics, and you wonder why we might call ourselves Fulcrum, well, if you think about a fulcrum, it's about manipulating a weight over a fulcrum. And our concept was we were going to try to bring down the high level of DUX4 expression and balance its expression in the muscles of patients. How would we seek to do that? We wanted to do it with an oral medicine that can be taken and that can systemically reach through the entire body and to every muscle that would be affected. It would, of course, need to be safe, and it would need to be effective. And we became very focused in patient interests, and we acquired tissues and cell lines from patients. We established novel methods to grow them in culture in our laboratories so that we could actually generate muscle from FSHD patients that were fused into myotubes and that were able to constrict in our tissue culture settings. So we established some beautiful models for screening. We undertook the screening of small molecule chemical moieties to see if we could find those that could reduce DUX4. We also took genetic approaches, and you heard a bit about CRISPR-Cas9 earlier, and we used that as a genetic tool for manipulating proteins that we thought could be important in DUX4 regulation. What we found were a series of molecules that could modulate DUX4. And we went on and we did a lot of evaluation of those molecules, and we came down to a clinical candidate. I also want to talk a little bit about the diagnostics question that was addressed to Rabi, to Dr. Tawil earlier on. We need patients to have a formal genetic diagnosis in order to be able to enter them into a clinical study. So if you don't have that formal diagnosis and you wish to be part of a clinical trial, I encourage you to get that. It'll be required before you enter. For Fulcrum, if patients come and don't have that, we will pay for that and we will help them to have that in order to be entered in. So we've come up with a molecule. It's called FTXA at this moment. We're still in preclinical stage, and we're planning for clinical evaluation of that molecule in FSHD patients next year.
We performed a screen where we specifically looked for molecules that could turn down DUX4, and that led us to what we call GBC0905, and I'm just going to tell you a little bit about what we know that molecule does, at least in our in vitro setting or in our culture dishes. So both in our model system as well as tissue that we have used from primary biopsies, so this is patients' biopsies, both FSHD1 and FSHD2 biopsies we got access to, we see that our compound actually turns off DUX4. And then it turns off all the downstream targets of DUX4. And that's really important because we believe that turning off DUX4 is going to be curative in FSHD. It also rescues the muscle cells from cell death. And then it does that in combination without having any sort of toxicity in our cultures, and as well as really not affecting the muscles. So what we get when we treat our muscle cells is we get healthy muscle that doesn't express DUX4 anymore. So, you know, I've put in here where we think our compound is playing a role, and it's really just turning off DUX4. So how we think that our molecule will probably work in patients is it will stop the progression of the disease. So I want to tell you a little bit about the status of this drug right now. We've recently just received orphan drug designation, and this is a designation given by the FDA that recognizes new therapeutics that are for rare diseases, and it's going to assist us as we move on to clinical trials. And right now, our timeline is that we plan on having interactions with the FDA and applying for a new investigational new drug application. And our target is to actually have this drug in the clinic by 2019, so basically the end of next year.
I'm from St. Louis University, and I'm working with a drug company called Ultragenyx Pharmaceuticals. to develop a drug to suppress DUX4 expression. And I want to tell you a little bit about focus on BET inhibitors and how we're using a mouse xenograft model for drug discovery. So the drug needs to get to the muscle and turn off DUX4. So this model is really enabling us to do quite a bit. So the first thing is pharmacokinetics. We want to make sure when we treat the mice with the drug that the drug not only gets into the system, but it goes into the muscle and it's there at a high enough concentration for a long enough period of time to have the effect on DUX4 that we hope it will have, which is suppressing DUX4 expression. So shown here is treatment with a tool BET inhibitor where we can suppress the two DUX4 targets, MBD3L2 and LUDX, with drug treatment with the mice. Interestingly, we found that when we treat the mice for 14 days with BET inhibitors, that we have an increased number of the FSHD cells there at the end of that treatment period. And so this is an interesting finding that we're actively following up on. And safety is also important. We want to make sure that our muscles develop normally while we're treating with BET inhibitors. MyH2 is a marker, a myosin heavy chain of late differentiation. And also, we can show that the BET inhibitors do not inhibit the generation of myofibers containing human spectrin. So this summarizes what I said. I just want to go down to the bottom. We're evaluating both repurposing candidates, which will be much quicker, as well as novel compounds for therapeutic development. We're comparing multiple mechanisms in this mouse model to compare because we only want to bring the best compound forward that's safe and effective.
Okay, I'm going to tell you very briefly about our work using CRISPR technology as a potential therapeutic approach for FSHD. So CRISPR is a gene editing system that's gotten a lot of attention in recent years due to the power and the relative ease of using it to modify the genomes of organisms. And most of the controversy surrounding this system has come from the idea of using it to modify the human genome, especially in ways that can be passed down to our offspring. This is not a new idea or a new concern, but CRISPR has made it much easier to do than ever before. So I think it's important to point out that what we're doing here is not this. We are not modifying eggs or sperm or embryos in any way. We are simply trying to use this system to correct muscle cells in FSHD. So the normal system uses a guide RNA, which binds to the Cas9 protein and recruits it to a specific location in the genome. So you can design your guide RNA to target, for example, the mutated dystrophin gene in Duchenne muscular dystrophy. And Cas9 will then cut the dystrophin gene. And if you provide a correct copy of the DNA, the cell's normal repair mechanisms will insert the new copy in place of the old. And this cutting system is what the vast majority of researchers are using to try to correct diseases. But this is not an ideal system for FSHD because, as you know, the disease locus is just one single repeat unit with hundreds of nearly identical repeats throughout the genome. And cutting your genome in hundreds of places is probably not a great idea. So an alternative system is CRISPR inhibition, and here the Cas9 protein is mutated, so it's incapable of cutting, and it's fused to a repressor protein. So now the guide RNA recruits a system that represses DNA instead of cutting it. And this is really ideally suited to FSHD, since we want to turn off or dial down DUX4 expression. And importantly, other similar repeats in the genome are normally repressed, so recruiting a repressor to these other locations shouldn't pose any unnecessary risk. And if the repression is stable, it can be inherited by subsequent generations of cells, but importantly without making any permanent changes to the genome. So in 2016, we published a proof of principle study showing that CRISPR inhibition can reduce pathogenic gene expression in FSHD. We used muscle cells from an FSH patient and showed that CRISPR inhibition could return the FSH locus to a more normal repressed state and significantly reduce expression of DUX4 in its target genes. And the important point from this study is that CRISPR targeting of the locus is conceptually possible, which was not a straightforward question. And we're currently testing CRISPR inhibition in our lab's mouse model of FSHD. Okay, so what are the challenges for CRISPR technology as a therapeutic avenue? There are some general challenges which apply to all diseases. Can we deliver the therapy to all of the target cells? Will all of the target cells be modified if we do? What happens if this system is recruited to other unintended places in the genome? This gets at the idea of off-target effects, which are understandably a serious concern. Can CRISPR components stimulate an immune response in patients, which would be a problem? And then, of course, can this system actually provide a long-term cure? And there are some FSHD-specific issues, which really are things we still need to learn about the disease. These are how many muscle fibers actually need to be corrected. Do muscle stem cells need to be corrected? And how much of a reduction in DUX4 expression will actually provide a therapeutic benefit? So as a therapeutic strategy, CRISPR is still in its infancy, but this platform is constantly evolving and being improved on because there are a lot of people working very hard on it, and it's potentially very powerful and very exciting.
The antisense therapy basically utilize or take advantage of natural mechanisms in our cells. So we didn't really invent anything. It's actually what our cells are doing. We just take advantage of that. So what happens is when there is a single strand RNA in your cell, and for example the DUX4 transcript, and then if you have a DNA strand that's complementary to it, in this case you can see the first letter A and then you have T at top, and then basically they're complementary with each other, and they actually can bind, they can hybridize, they can bind tightly with each other. And in that case, for example, for therapeutic purpose, we can put a small stretch of this DNA molecule, and then if we can deliver them into the cell successfully, then they can actually search for the sequence that's actually complementary to it. In this case, they actually can achieve a very specific target of the RNA they want to reduce or knock down. So they, in a way, one of the advantage of antisense therapy is actually to have a more specific knockdown. So you only knock down DUX4, not something else by accident. And when that happens, there's an enzyme inside your cell. Here, RNase H actually will recognize this DNA and RNA duplex, and then they will degrade the RNA that's associated with your antisense molecule. So that's basically how it works. There's a lot of practical problems. For example, if it's a natural form, it's not very stable, and it will be degraded quickly. So there are additional modifications that's chemically to the natural form of RNA. So there's one example, that's what I'm going to talk about later. It's LNA. So you can see there are two different arrows, red arrows there, actually tell you how it's modified. And you can see one of oxygen is replaced with the sulfur, and then the other one is actually a different linking pattern. And then basically these improve the stability of this molecule, and it actually can stay in the cells longer for therapeutic purpose. So you don't need to have it every hours or something like that. So for the study, we actually use a mouse model. This is where we are at the moment from Peter Jones' lab. And the animal model actually expressed DUX4 at very, very low level. It's so low, it doesn't even induce those downstream changes. However, these animals actually show muscle weakness and some pathology we see in muscular dystrophy. So we thought this probably is a good model to start with, because we want to see, even with this low expression of DUX4, whether we can do anything about it. So we first try to see if we can see that compound get into the muscle. So this is actually a fluorescent labeled compound, and then this A is a control, but you can see B and C actually they can get into the muscle tissue. Okay, and for the next several slides, I just want to show that after the first experiment, we actually did subcutaneous injections. So we delivered them several times within a period of time, and then check whether it can reduce DUX4 in vivo, so inside the muscle. So there are two different muscles collected here. One is the quadriceps and the thigh muscle, and the other one is gastroc, the, how do you call that, this part. And as you can see that the first bar is actually from the wild-type sibling. These animals do not carry the DUX4. They don't express DUX4. And then the second bar is actually from the animal that expressed DUX4. And the third one is actually after they receive the treatment for 6 injections every other day. The most recent is a longer trial. We want to see if we can improve muscle function, make them stronger. So this basically just show you that in this trial, we can also knock down DUX4 to even greater extent. And this actually just to show that it actually improved the grip strength.
Okay, so I mentioned earlier that I've been focused on gene therapy for many years. I'm only going to tell you about one of the gene therapy strategies we have going in the interest of time. But I do want to mention and spend a minute or so just telling you about gene therapy. It is not it's not science fiction anymore. We actually have the first approved gene therapy on the market now. FDA approved it. It's actually a treatment for blindness. I'm going to show you that because I think it's exciting and it helps inform how we're thinking about treating FSHD with gene therapy. So a couple of the advantages of it are that, well, the major advantage of using gene therapy to treat any disease is that in general, it is a one-time treatment. So we're delivering genetic material that should, if it works correctly, correct the disease. And the way we're doing that is to use a modified virus. So viruses have had millions or hundreds of millions of years to evolve and figure out ways to enter tissues. And so they know how there are certain viruses that know how to safely get into muscle, and we could use those to deliver genes that we want to use for therapy. Okay, so I've been focused on gene therapy for many years. I'm only going to tell you about one of the gene therapy strategies we have going in the interest of time. But I do want to mention and spend a minute or so just telling you about gene therapy. It is not it's not science fiction anymore. We actually have the first approved gene therapy on the market now. FDA approved it. It's actually a treatment for blindness. I'm going to show you that because I think it's exciting and it helps inform how we're thinking about treating FSHD with gene therapy. So a couple of the advantages of it are that, well, the major advantage of using gene therapy to treat any disease is that in general, it is a one-time treatment. So we're delivering genetic material that should, if it works correctly, correct the disease. And the way we're doing that is to use a modified virus. So viruses have had millions or hundreds of millions of years to evolve and figure out ways to enter tissues. And so they know how there are certain viruses that know how to safely get into muscle, and we could use those to deliver genes that we want to use for therapy. Okay, so can you show the first video, please? 1A. So I mentioned that the first approved gene therapy is on the market now. It's a product called Luxturna. This is a treatment for blindness. And I'm showing this for two reasons. One, again, to demonstrate that gene therapy is a reality now. And the second, because we're using a lot of the similar tools that they're using here for blindness. So what we have here is a little boy who's blind. He's been treated with the gene therapy in one eye. And what they've asked him to do is they've covered the treated eye, and they've asked him to walk through a maze that has black arrows on the floor. And so you can see that he can't navigate. And if you heard this, you could hear the sound, he would say, "This is hard, I can't see anything." You can sort of see that type there. But basically this goes on for 3 minutes, and he never makes it through the maze. Can you shut this one off now? Put on the other. Here we go. Okay. I mean, to me, that's amazing. That's miraculous. Okay, so the point is that we've been thinking as a field about gene therapy for 40 years, and there have been a lot of hurdles, a lot of setbacks, and now we finally have the first treatment on the market. There are still a lot of hurdles that need to be cleared to actually make this feasible and make it widespread. I mean, they're treating the eye versus, an eye is a very small space versus an entire human being's musculature. So there are hurdles, but we are using the same types of viruses they're using here, and we're able to deliver these viruses to muscle. And so I'm just going to show you very quickly because we're running out of time here. One of the things we're doing now in mice, so without spending too much time on mechanism, I'll just say that we can clone these small RNAs, similar to the antisense that Dr. Chen mentioned, but we can express these from a virus, and we can package those inside the virus. We sort of trick it into thinking that it's delivering its own genes, but instead of delivering its own genes, it's delivering our payload. We can deliver that to a muscle either by intramuscular injection or actually through the bloodstream, and it will find the muscle. This particular virus likes to infect muscle. And this particular virus also, it's called adeno-associated virus, is not known to cause any diseases in humans, so it's a benign system. And when we deliver this, what we call a microRNA, this microRNA is designed to knock down the DUX4 gene. When we packaged our microRNA system that knocks down DUX4 into this virus and delivered it to mice, I'll just focus your attention down to I guess it would be the bottom left. You can see this is the same mouse now. One muscle, so this is the calf muscle. One's been treated with our therapeutic vector. The other one has not been treated. And I think you can just look at the low power shot there, the two muscles. The blue indicates muscle damage. And so the untreated muscle has widespread muscle damage, and we can completely protect the mouse from muscle damage with our gene therapy vector.